All right, sweet. So mutations. We're going to define mutation. The biggest giveaway that I tell my kids is always keep the M's together. Like I do this all the time. I don't know why. It's my way of word association to help me remember. So in the past, I've told you peptide protein, keep the P's together. Here we keep the M's together. Mutations are mistakes. And sometimes we make mistakes in our own lives that are bad and there's consequences. And sometimes we make a mistake that ends up being a blessing, right? So we're going to talk about how mutations aren't always bad things today. And we're going to have, or at least understand the effects on our traits, which are our genes, and then chromosomal mutations and what that means, right? So, of course, my girls, the amoebas, are going to start it off and then we'll finish off talking about it.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random, but there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched, insertion, which means an extra base or bases are added in. There is also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have three bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but The idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication. But also, there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have half the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit two copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. While unfortunately there is not a cure for this disorder yet, the good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits one copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Ooh, stay curious. Okay, that's enough amoebas. All right, so a lot of the stuff that they said is just going to be repetition when I go through it. This slide presentation is already in today's folder. I'm going to post the video lecture of it, which is what I'm recording right now. And I've created a one-pager of if this is all that you learn, you'll be okay. And so we'll post that in the folder as well. So what is a mutation? Again, keep the M's together. A mutation is a mistake that has happened. You can either insert the wrong one, skip a base, or the big three are this. You add an extra one that shouldn't have been there. You accidentally leave one out, so you're missing one, or you do a switch where you put the wrong one in, right? And so all of these mutations, right? The root word of mutation comes from the Latin word that means to change. So again, if we go back to like the beginning of the year and why we use Latin, it's because it's a dead, unchanging language. And so therefore, we don't have to worry about any particular country coming in and saying like, oh, that's ours, so we get credit, because nobody claims Latin as their native tongue, right? Mutations are passed down, so they're heritable changes. You inherit them from your parents. So if your parents have a mutation, and again, if it's in the code that is in the sperm or egg, then it'll get passed down from one generation to the next. And having just come out of the DNA unit with the scientists and Oswald Avery being the one that basically coined that DNA is the genetic information passed down from one generation to the next. Okay, so there's different types of mutations. There's actually two. There's going to be these gene mutations, which are going to affect traits. So, for example, your hair color, your eye color, stuff like that. My father in law is a white German man. That's why Gottschalk is that my last name, right? It's a German last name. So he's your typical German man. Like he is blonde and he has one green eye. And the reason I say one is because he actually has that God, what is it called? It just slipped my mind. He has that mutation where he has like one light brown eye and one green eye. Um, and that's a gene mutation. So it affects one particular gene, one trait on his body. So your genes are inside you, they're in the DNA. And the genes code for proteins that make up our traits. So that could be your hair color. This could be your skin color. A lot of kids don't actually think that skin color can be a mutation, but when you think about like albinoism, that's a mutation that's affecting a specific gene that's affecting your pigmentation in your skin, right? We can also have chromosomal mutations, and those are going to affect the entire chromosome. Remember your X's when we were doing our cell cycle, the X's are our chromosomes. And so we are going to talk about those a lot more once we get into genetics and we'll learn how to name these mutations and such. But for right now, we just need to understand kind of the difference between gene and chromosomal. Now, mutations are more prone to happening with sexually reproducing organisms, right? So either your plants or animals. And the reason being is that now we're opening up a can of worms with meiosis, which we haven't discussed yet. And so it's one of those things where we don't want to get too deep into it because we don't want to confuse. But if we are going to now combine mom and dad's genetic information to create you, you are going to go through this process, right, of being able to take mom and dad's genetic information and then cross it, right? And so this crossing over concept is where now they basically splice mom and dad's and you become a combination mixture of both. It can happen in asexual reproducing organisms such as protists and bacteria. There is going to be some sort of environmental component when it comes to those asexually reproducing. I know in the past we've talked about transformation when we talked about Griffith and the mice. Y'all remember the mice experiment where we had the bacteria that was good, injected it, they lived. Bacteria that was bad, injected it, they died. We heat killed the bacteria, so we boiled it and it was no longer alive. Injected it, they lived. And then when we mixed the two that lived, it actually killed because the bacteria transformed. So a lot of different components, but really all that we're keeping in mind right now is a mutation is a mistake. Mutation is a mistake, right? We talked about pigmentation being a gene mutation where it affects a trait. Here you see this elongated flower tissue. With the gene mutations, there's going to be three main types of point mutations. There is a substitution, an insertion, and a deletion. And the thing about these last two is that these are actually called frame shift mutations. And let me show you kind of what that means. When we are making copies of the DNA, when we are replicating, we are actually in S, synthesis of interphase, which is the second step of interphase. Interphase has three parts. G1, S, G2. And we talked about how in the G's we're growing and S we're making. And what we're making is we're making a copy of DNA. If we're making a copy, then we can potentially make a mistake. Here is my original sequence. On the top in black, I have my DNA. How do I know that's DNA? Well, I see T's, right? In my second row with the light blue, this is my mRNA. How do I know it's mRNA? Well, there's U's and they're in groups of three. They're in codons. This is going to then give me an amino acid. This is the translation bit. And so we've already done this. We know transcription, we know translation. When we have these mutations, these point mutations, you'll literally be able to take your finger and say, boom, that's the mistake. So here where we used to have a C in the original DNA sequence, it's actually been copied wrong and now a T was in its place. And what you can see is that now that one point mutation is going to give you, this is a substitution because we changed the C for a T. For substitutions with me, I always think of a sports analogy because I played sports growing up, right? And so if you have a sub, there's still going to be a shortstop. It's just, it may not be me anymore. They may have replaced me with somebody else, right? So here you have this different amino acid that was now pulled, and obviously that's going to give us the wrong protein sequence. Insertion and deletion are our frame shift mutations because everything after the insert, so we added one, or the deletion, we deleted one, is going to be wrong. So here, from this point forward, everything that is coded, and that's why you see them in red, is wrong. The reason that they're called frame shift mutations is because they actually shift our reading frame. Since we read mRNA in groups of 3, if we add one or delete one, everything's either going to shift to the left or shift over to the right. Every single amino acid following the frame shift mutation is going to be incorrect. And because all of my amino acids are incorrect, and amino acids are the building blocks of proteins, the protein that I make won't be able to do its normal function. An example of a frame shift disease is going to be Tay-Sachs. Tay-Sachs is going to start to develop symptoms around 6 months of age. So my daughter Blake is 3 months. She'll actually be 3 months and 2 days. And there are certain things that as parents of a young child, and if you guys have like little baby cousins or baby brothers and sisters, you know that like every month there's something that you should be looking for that these babies are now able to do. The first month, it's kind of boring. I shouldn't say boring. It's very tiring, but the babies are kind of boring being around them because they're like a sack of potatoes. Like they don't do anything. They just like lay there like Hannibal Lecter all wrapped up, and they just eat, poop, and sleep, and they don't do anything else, right? So she is now in this like really fun stage where if you've ever seen the Grinch and like, you know, he like the baby Grinch is eating like the Santa plate and he like moves his little head. So she's doing that. She has like the little bobblehead stage right now where she's trying to hold her head up and she's trying to be really independent. Um, and she's kind of smiling back at us and she's cooing. So a lot of vowel noises, like you'll see like ooh, ah. And so it's starting to get fun. The boys are now actually interacting with her and they're like, oh, I guess she's cool. We can keep her. Well, around 6 months of age, a baby, right, should be able to sit either with assist. So, like, you know, you could put a pillow behind their back and they could sit up independently on the floor. They should be able to roll over from front to back and back to front. And they should be able to, babies have this natural rooting reflex. So if you touch their palms or you touch their feet, immediately their fingers or their toes curl in. And that's why it's really cute and it's really easy actually to get a picture of a baby holding your finger because all you got to do is put that finger near their palm and they'll immediately close their fingers down on it to hold on to it. So what they ask you to do with babies is to grab their hands, and since they immediately want to make a fist, you pull the babies up, and this builds up their core strength, their neck strength, etc. So around 6 months, when babies should be doing this sitting and this rolling, what babies with Tay-Sachs disease actually start to experience is that their nerves, instead of making connections within these synapses, their neurons, they actually start to deteriorate. And so what that means is that they start to accumulate fat in their brain, and the fat actually starts to suppress all of this tissue. And so these babies, they actually go blind, deaf, and they lose the ability to swallow. And so the only prognosis for this type of disease is it's terminal. They're going to die from it. Majority of patients who have Tay-Sachs disease won't live to be more than 4 years of age. And typically when you see those cases of the babies that get to be 4 years old, it's because they're on all kinds of, extensive measures. They're on feeding tubes and they're on a vent or they're on all types of different machines to keep them alive, right? Because any other way their body would not be able to do it. So it's really sad when you think about it. And that's because one tiny letter was added and that threw everything else off. Now, a chromosomal mutation is going to be when we have an extra chromosome or we're missing a chromosome. And so the most common um, type of chromosomal mutation that you guys may have been, exposed or familiar with is going to be trisomy 21, which is Down syndrome. And individuals with Down syndrome are able to go to school, go to college, get married, have jobs, drive a car. They're able to be very independent individuals. And so, whereas maybe 50 years ago, um, the diagnosis of a child with Down syndrome may have seemed like it was going to be the end of the world. Now we are very educated and there's a lot of resources for individuals, right? And all of the individuals with this trisomy are going to share some common characteristics, right? And so it's one of those things where you can recognize that somebody has this chromosomal mutation by looking at them. There are other mutations, and when we get into genetics, I'll talk about them, where Edwards syndrome and different types of trisomies will actually lead to they're terminal, they're fatal. So these babies will not, if they survive till birth, they won't survive long after. Um, and so like I said, once we get into genetics, we'll talk about it a little bit more. But there are four main types of chromosomal mutations, and here is the best part about it is that they tell you exactly what is happening in the name. So in a deletion, we're getting rid of something. In a duplication, we're making a copy. The one that the kids struggle with the most is the inversion. And that's because we are taking the reverse, and that's what the amoeba sisters told us. And then the translocation. Trans means change, location is going to be, and so here you see that the locus of the gene has been, or this part has been added and moved into this particular chromosome, right? So you had your original that went from A to F. You can see that the B is missing in the deletion, that there's an extra one in the duplication, that B and C were switched in inversion. And you guys have heard about taking the inverse when you guys do fractions and math and stuff like that. And then here again, the translocation, this GH moved into this location. Now, more often than not, we like to think that a mutation is going to be negative, because when we think of a mistake, it's typically has this negative connotation to it. Sometimes because there are 64 different combinations of codons, but there's only 20 amino acids, sometimes you'll make a mistake, but it'll still code for the same amino acids. So you'll still make the same protein, and this will be known as a silent mutation because it goes unnoticed. Sometimes you'll have mutations that are actually beneficial. And so to give you a couple of examples is that there's organisms that have developed an evolutionary advantage because they have either become resistant. So there's bugs that are pesticide resistant. So the spray that we put out to kill them is actually not going to do anything to them. There's individuals that are immune to HIV. There are people who have bigger bones that keeps them from breaking. So these are all things that are, sometimes, you hear, oh, I'm big boned. And like, yeah, it's not a thing. It is a thing. And then sometimes, of course, what we're more prone to going to when we think about a mistake is that it's a bad thing and it's negative. Now, if it wasn't for mutations, us as humans, we would lose the ability to evolve, which means to change over time. And so because evolution is a characteristic of being alive, we would not be able to continue evolving if it wasn't for these mistakes that occur. Now, the bad. Let's talk about the bad first, right? Because we just got out of cancer and the cell cycle, it makes sense for us to talk about it. The cell cycle is regulated by these little signals, right? We call they're cyclins, but they tell you, yes, make more cells because we're either growing or repairing, or stop, we've done our job. When the stop sign breaks, cancer just makes cells that are not needed. They accumulate in clusters we call tumors, and then that's when we diagnose. So that's obviously a negative mutation. The other type that they talked about in the video was the sickle cell anemia. And so your red blood cells are actually going to be transporting nutrients and oxygen, and they should look like these little circle smarties, little hockey pucks. But in sickle cell anemia, they're going to look like these little slivers, and therefore they're not going to be able to carry as much oxygen. And so therefore the body's going to be deprived of oxygen. And so it's one of those things where you treat the symptoms. There's no real cure for it. And blood transfusions are typical and common in sickle cell anemia patients. Now, the positive. We are going to have, like I said, mutations that make insects resistant to pesticides. We have individuals who have an increased bone density, so it makes them less likely to have fractures. And then we're going to have individuals who are resistant against certain viruses. And this is one of those things that is super interesting to scientists because they want to study the 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 genome of those individuals that have this resistancy, because then if they're able to pinpoint what triggers the body to combat this virus, then we would hopefully be able to use that to cure people with it, right? And then last but not least, polyploid plants, because mutations can happen in both animals and plants. We have to think about us as consumers. If you've ever gone grocery shopping with mom, we love to get the most bang for our buck. And if you go into a grocery store and you go to the produce where there's the limes and tomatoes and all of that, people are picking through the veggies, trying to get the prettiest ones, trying to get the biggest ones. Um, and so for farmers and animal breeders, they're going to make use of these good mutations so that they can cross these organisms in their sexual reproduction and produce plants that are going to be bigger, um, larger, stronger, etc. Because if we are both farmers out in Poteet and Jaden is growing strawberries and I'm growing strawberries and we're trying to sell them to HEB for the exact same price, it's now going to come down to quality. And if my strawberries are twice as big as Jaden's, it's more likely that I'm going to land the job with HEB because I have something that the consumers are going to want, right? Now, like I said, I will be posting the one sheet page of if you don't learn anything else, this is the bare minimum of what you need to know onto today's folder along with the video recording of the lecture, right?